Okay, let's finish up the analysis piece. We have a few slides left, and then we'll move on to the next topic. Trying to remember where I was last time. I think we covered this and we stopped at Maldi. Correct? Did I cover Maldi or not yet?
Not
Nobody
yet. I don't
remembers.
think so.
Okay. All right. So I don't have a recap questions since I was supposed to finish this lecture last time, but do you have any questions or anything you want to discuss before continuing with and continuing and finishing up structural analysis? Any questions from before? Okay. All right, so last time we ended up talking about using size exclusion chromatography to characterize proteins, looking at polymerization, looking at hydrolysis, looking at molecular weight distribution, and so on. So we can also use microscopy. I don't know if I talked about that last time, but SEM looking at morphology, or also we can do CLSM, which is confocal laser scanning microscopy, where we can stain the constituents of the sample. Proteins can be stained green, and we can monitor I'll give you examples later on CLSM pictures and images, but you can look at the three-dimensional network of a protein in a cheese sample, for example, or in an emulsion. You can determine where the protein is located at the interface. So as at the interface compared to the continuous phase. So microscopy is another tool of looking not just at aggregation, but also distribution of the protein in a particular system. All right, so MALDI. So MALDI-TOF, which is matrix-assisted laser desorption, ionization, time of flight, mass spectrometry. So it has many applications, and I'll show you a few examples, but commonly it's for identification of a protein or a peptide, determining modification sites. I'll give you an example about that. Looking at enzymatic modification and what peptides are generated, potentially giving us sequence of the peptides that are generated. So let me just briefly then, again, there is a sheet on MALDI TOF in the reading material, but basically you have a weak organic acid as your matrix, and you have your sample with the matrix, and then a laser is focused on where your matrix and the sample is, and the matrix absorbs the light and then causes ionization of your protein without fragmentation. And then the solvent is evaporated, and the energy that is generated propels the ions into a time of flight tube. So, and the ions travel based on their size. So the smaller they are, they reach the detector sooner. That's why it's called time of flight, because it's based on how much time it takes to reach the detector, and it's correlated with the size of the molecule. So oftentimes you do a 2D gel, for example, to purify as much as you can the protein. So you run it on one-dimensional gel, and then you run again your sample on a second dimension. So you get your proteins separated. Sometimes you take a protein off of that gel and treat it to be analyzed by MALDI, or sometimes you take it off of a chromatographic separation. You take a fraction that is pure protein, and then you can subject it to MALDI-TOF analysis. So what precedes the analysis is a trypsin hydrolysis, so that you can break the protein into peptides, and then you can potentially has better chances of flight, ionization and flight and detection. And then you can piece together the information that you get from the peptides to determine the original identity of the protein. So you can use MALDI also to do something we call ladder sequencing, meaning you would have cycles where you hydrolyze the protein or the peptide where at one amino acid at a time, and it goes into the MALDI. And then you see here, for example, the difference between one signal and another is one amino acid. Then you can, by ladder sequencing, you can sequence the entire peptide by having a cycle, multiple cycles of hydrolysis and breakdown of one amino acid at a time. Here's an example where we used MALDI in a research in our lab a few years back. We were looking at glycating whey protein. So meaning covalently linking a carbohydrate to whey protein. And we determined that this type of glycation resulted in a rigid structure that resisted denaturation. So we wanted to see what changed, or where is the site of that glycation occurred that resulted in this rigidity of the structure. So we got our protein that was glycated, and we subjected it to trypsin hydrolysis and also to dextranase, an enzyme, so that we break the long chain of carbohydrates so that it can easily fly and can be detected. So, and we noticed, so we ran the protein that was not glycated and the protein that was glycated, and we noticed few new peaks that were not there originally. And we determined the sequence, obviously, and determined the lysine that got blocked by the carbohydrate, by being covalently linked to carbohydrate. And the location of that lysine was very unique in the structure of whey protein that prevented the protein from being easily denatured and susceptible to unfolding. So that was a very nice piece of information. We were able to link the change in the structure and the location of that change that resulted in structural rigidity that explained the stability of that protein we produced. LC-MS mostly is used to determine a lot. It's used for so many different applications in proteomics world, but it helps a lot in determining enzyme selectivity if we are looking at the hydrolysis using different enzymes. And it can also be used for identification of proteins from different sources. So, for example, we used it in studying intermediate wheatgrass, the proteins from intermediate wheatgrass compared to the proteins from wheat. And we were able to look at similarities and differences using the LC-MS approach. So here's an example where we used LC-MS several years ago. We were looking at producing a hydrolysate from soy with bioactivity, specifically antihypertensive activity. So it was shown in literature that peptides from one of the proteins, beta-conglycinin, for example, produce bioactive peptides that are more potent than the peptides that will be produced from the other protein in soy, which is glycine. So IC50 is the concentration of the inhibitor of antihypertensive enzyme. So the inhibitor, so this the smaller the value, that means you can get 50% inhibition. So better inhibition. So the smaller the IC50, the better the inhibition is, which is the concentration at which you get 50% inhibition. Anyway, so the point was there were literature that says that we need to isolate these proteins and produce the bioactives from these isolated fractions. What we tried to prove here is instead of having to fractionate these separately and add excessive amount of time and to process and produce a fraction that is rich in one protein versus another, and then hydrolyze it to produce the peptides that are antihypertensive, we said we used LC-MS to show that whether we hydrolyze the fraction that is purified or we hydrolyze the same protein within the isolate before fractionation, we're going to get the same output. So what we did is we isolated the fractions and hydrolyzed them by the same enzymes. And we looked at the peptides that were generated. So we used LC-MS to determine the peptides that were generated upon hydrolysis, whether hydrolyzed as an isolate or hydrolyzed as a pure fraction. And we found that you don't really need to fractionate to get the same sequence of peptides, especially if you use selective enzymes. So if you use selective enzyme, pepsin is selective for glycinin and papain was selective for beta-conglycinin. We were able to prove that the peptides generated are the same, whether it's pure fraction or not. So LC-MS was a powerful tool for us in that regard. This is a more recent study that we just completed, and we used LC-MS. What you're looking at is a map, a peptide map, which is a very cool way of looking at peptides generated from certain enzyme. So here we were looking at differences in peptides generated using trypsin and a bacterial enzyme, the protease M. So two different enzymes, and we looked at the hydrolysis pattern towards beta-lactoglobulin. So what we see here, we see that there is selectivity at the lysine, which is not a surprise for us that most of the peptides generated were at the lysine location breakage, because that's trypsin, lysine, and arginine are the locations for trypsin hydrolysis. So, and also we looked at specificity of the other enzyme, determine where the peptides were generated and where the enzyme cut. So what you're looking at here is basically a map. So this is the sequence of beta-lactoglobulin. So it starts here and it ends here, 162 amino acid. So we know the sequence. So the computer has the sequence, and we were able to analyze the peptides in select fractions we isolated by size exclusion chromatography, and we were able to find out where these peptides are coming from the original sequence. It's pretty cool and powerful technique. Another thing we were able to look at is using bioinformatics, looking at the hydrophobicity of the peptides, hydrophobicity versus hydrophilicity of the peptides. So that was really cool because we were able to get something called Gravy score. So a Gravy score is a score of grand average score of hydrophilicity or hydrophobicity. So basically, any score above 0 means that the peptide is hydrophobic. and scores below 0 means that the peptides are hydrophilic. So here you can see that trypsin hydrolysis resulted in a balance of some hydrophobic and some hydrophilic peptides in different fractions that we collected from the hydrolysate, whereas the protease M enzyme from bacterial origin generated a lot of hydrophilic peptides. Very few scored above 0. This kind of proved our point for this particular research is that we can use an enzyme to enhance functionality, for example, without producing hydrophobic peptides that might potentially be bitter. So you can do a lot of different things with LC-MS as well as bioinformatics that comes as a tool to analyze your peptides. Last thing here is NMR. NMR is also used to determine the tertiary structure of the protein based on the signaling and spaces of protons in the sequence of amino acids. So that is another technique. We don't use it often. We don't use it in our lab, but it is a technique used for protein elucidation of protein's tertiary structure. Okay, so far we looked at molecular interactions, molecular size, denaturation. We will be talking more about denaturation later on, surface properties. Let's talk a little bit about amino acid composition and sequence. So why do we care about amino acid analysis? So basically, we determine composition for nutritional quality, but we also want to understand any new isolated protein. We need to understand it. We need to know its composition of amino acid and also the sequence of amino acid. And it helps determine another way of determining molecular weight by adding up the amino acids, individual molecular weight of amino acids. You can get the total molecular weight of the protein. So how do we determine that? It's basically the most common method is hydrolyzing with a strong acid for 24 hours. So 6 normal HCl, you break down all the peptides, you end up with free amino acids, and the amino acids get separated by reverse phase chromatography, and they get, or ion exchange, but mostly it's reverse phase chromatography. And we use, we quantify by reaction with an anhydride or OPA reagent. So we can measure absorption or we can measure fluorescence, depending on the availability of our detector. The issue with using strong acid is we get some amino acids, they get destroyed, like tryptophan, for example. We cannot detect it that way. So oftentimes, in order to get the complete amino acid sequence or composition, we have to do multiple ways of hydrolysis. For tryptophan, we can do NOH hydrolysis by alkaline, that is. And also we can, for cysteine and methionine, we have to use a specific reagent to allow their detection. So oftentimes there are 3 different ways we hydrolyze, and so that we can get the complete amino acid composition. The sequence we can do, like we said earlier, we can do by MALDI, the ladder sequencing by MALDI, but also there is a common technique called Edman sequencing, and it's also known commonly as N-terminal sequencing. But it's called Edman based on the person that developed this method. So it's basically a reagent react with the first amino acid from the N-terminal, breaks the bond, and you end up with a compound that gets detected or absorbed light at 270 nanometer. Every cycle, the reagent reacts with the next amino acid. So it reacts with one amino acid at a time, and then then the amino acid is projected to a chromatography in reverse phase chromatography, and then you get your amino acids in sequence based on the cycle of the reaction. So in order to be able to do N-terminal sequencing, we need a really very pure protein. And oftentimes one dimensional gel is not enough. We do two-dimensional gel, and we excise the peptide or the protein out, treated with the reagent, and then subjected to chromatography separation. And often the peptide is taken off of polyvinylidene fluoride, which is basically a membrane where we transfer the protein from a gel onto a membrane, and from there we take the protein and do the analysis. Pretty tedious, takes a long time, but it's pretty robust as well. But the protein really has to be very pure. Otherwise, if you have other proteins in there, you won't get a correct sequence of that particular protein of interest.
Pam, I had a question.
Mm-hmm.
Example where you showed that Whey has 162 amino acids, and if we were to use N-terminal sequencing to get to the sequence, would we have to subject it, like react it with the reagent 162 times because it analyzes one amino acid?
Yeah, usually you'll have a cycle for every amino acid, yes.
Okay.
And each cycle is timed, and there is a specific equipment that you would use to do that cycle, the timed cycles to get one amino acid at a time.
And if you're dealing with a protein, you don't know how many amino acids are in there, so you just keep going
Until there are no more. Yes,
Okay.
yes, absolutely. Okay. All right, so identification, oftentimes we use ELISA and Western blots to identify presence of a foreign protein that's not supposed to be there, but it's there, like horse meat in a beef patty, for example. So we use ELISA with specific antibodies for a protein that we're trying to determine its presence. Um, so most common is the sandwich ELISA and the indirect sandwich ELISA. So most of you might have seen this before in previous classes, but basically you have a capture antibody, and then if you have the target protein, it will bind to it. And then we have the antibody that would be reacting at another epitope to sandwich the protein. And most commonly, we use indirect ELISA because it's easier. We have reagents already to have an antibody to the antibody. So if we're using human antibody, for example, we can get antibody to a human antibody that is already labeled with the enzyme that will convert a product to a color. So that's the enzyme labeled immunosorbent assay, because then that's the enzyme that is labeling the secondary antibody. So I won't dive into that. Immunoassays can take a whole lecture describing immunoassays, but basically they are used to determine presence of either an allergen or a foreign protein that's not supposed to be there, or a GMO protein, for example, as well. So we use the ELISA assays, which is more quantitative. We can get quantitative data if we have standards. Western blot is qualitative, and we use it to screen a lot of samples. Basically, if you have a mixture of proteins and then you run them on first dimensional electrophoresis, then a second dimension electrophoresis, and you blot them onto a membrane, and from there you react with your primary antibody, that is the capture antibody. If you have that protein of interest, it will bind to it. Then you add antibody that is labeled, which binds to the antibody to the antibody, and then you get a color. And if that protein is there, you see the spot on your membrane. So we've used this technology to screen soy protein allergy. And we used, for antibody sources, we used serum from individuals that have allergenicity to soy. So that means the serum has antibodies that are specific, that can specifically react with soy protein. So what we did here is a gel, regular gel, and we blotted it on onto a membrane, and we incubated it with the antibodies from the sera, with the sera that has antibodies. And then we were able to see all the proteins that reacted, that are still immune reactive. So basically we were treating the protein by hydrolysis, and then we were treating the hydrolysate by glycating it, adding a sugar to it to see if we can even reduce the immunoreactivity. And we were able to see that certain treatment resulted in a much reduced immunoreactivity compared to the original protein. So when we're doing just screening, that's a very good method, quick, and gives us information about changes in the protein that might have resulted or caused a change in immunoreactivity. Last example I have here is oftentimes when we want to characterize a specific protein, we really have to go through elaborate ways of isolating it. If we are looking at characterizing bioactivity of a potent peptide within a mixture of peptides that we produced by enzyme hydrolysis, we really need to go through a sequential separation procedure to be able to get a purified peptide that we can sequence and we can characterize for its bioactivity. So for example, if I start with a hydrolysate, the first thing I want to do is I want to remove the small peptides that are usually your potent peptides that are bioactive, remove them from the larger polypeptides that are present in the sample. And we often do that by ultrafiltration. So take everything less than 5 kilodalton or less than 3 kilodalton. From there, we can do liquid chromatography, ion exchange followed by size exclusion, or size exclusion followed by ion exchange. It doesn't matter as long as it's two-dimensional. So what we do here in this example is we run the peptides, let's say, on ion exchange, and we take each fraction and we measure bioactivity. Then we take the fraction that had the highest bioactivity and subject it even to another chromatographic technique. And then here, for example, size exclusion. So I take that fraction that was bioactive from the ion exchange, and then I separate it further, and I run my assay on each fraction. Then I take the fraction that is most potent, and from there I can do reverse phase chromatography to separate even more until I get a purified one peptide that is purified. And it is the most potent. And in order to characterize it, I would take this fraction that is most pure now because it went through at least three liquid chromatography separations. And then I can do either MALDI to determine sequence, or I can do N-terminal sequencing on it. So that's just an example of how do we characterize, for example, a potent peptide. We have to purify it first, and then identify it and determine its sequence. All right. Any questions at all all about analysis. Anything you'd like me to go back before changing subjects? Okay, so we're gonna go out of here. Stop share for a second. And share the next one. Okay, so we're moving into protein denaturation and functional properties topic. Let's try to answer this question. The following is true. A decrease in enthalpy results in a decrease in entropy. Breaking of few bonds result in a decrease in entropy. A decrease in enthalpy results in a substantial increase in entropy. You want to remember what enthalpy is and what entropy is. Who will volunteer and tell me what enthalpy is? Does anybody remember what enthalpy is?
The heat capacity.
Energy.
Say that. Regina, say that again.
Is it enthalpy is the energy contained and then entropy is the chaos?
Yes. So enthalpy is the energy contained in the bonds. So when a protein is formed and it exists in an aqueous environment, it has so much free energy that it's not stable. So it starts to fold and form interactions, hydrophobic interaction, ionic, hydrogen bonding, van der Waals, covalent, until it forms its final structure, either a tertiary final structure or a quaternary final structure that contained the energy. Okay, entropy is the chaos, the opposite. So if we break few bonds, we are releasing energy and then we are releasing a lot of chaos. So basically the answer here is a decrease in enthalpy due to breakage of few bonds can result in substantial increase in entropy, which results in negative delta G, which is spontaneous denaturation. Okay, we'll talk more about that here in a little bit. So denaturation induced by heat can be enhanced in moist conditions, can be reduced in the presence of sugar, can be enhanced in the presence of salt. What do you think? Okay, some people are putting answers in the chat, where is my chat? Why can't I see it? No answer? I can't see my chat for some reason.
Most people are saying F.
Most people are saying F. Okay, why can't I see my chat? I don't know why I don't see the chat box, which is sad. Okay, so can be enhanced in moist conditions. That is correct. So when the higher moisture content, the denaturation temperature is lower, and we'll talk about that later. I'll show you in the figure later on. So water enhances denaturation of the protein. Can be reduced in the presence of sugar. Yes, small sugar can stabilize the protein, bind lightly with the protein on the surface, allows for more water interactions. Can be enhanced in the presence of salt. Certain salts result in enhanced denaturation by binding very strongly to the protein. So depends on the type of the salt. So the answer, because it says can be, all of them are correct. Okay, and we're going to talk about each one of those individually, of course. All right, so bringing you back to the map of where we are. So what we covered so far is the protein structure and the intrinsic factors that impact the protein structure. And then we covered analysis of the structure. This is important because what we want to do next is look at how the protein structure changes upon denaturation or modification. And then we talk about the functional properties, and we're going to relate structure to function, okay? At every point, understanding the structure, that's why we did the analysis. We need to understand the structure, and we need to know how to analyze the structure. And we're going to learn functional properties and how we analyze functional properties and how we relate structure to function. So the next couple of lectures, maybe today and next time, we're going to talk about denaturation and then functional properties. And then I'm going to give you examples of linking structure to function. For every function we talk about, I link structure to function by examples as we go. Okay, so here's where we are at. So we're going to talk about denaturation next. All right, so foods are processed. We rarely eat stuff raw other than fruits and vegetables. So processing is required, and this is not something new to you, not the news. We process to inactivate undesirable enzymes, to make the food safe by killing microorganisms, pathogenic and spoilage microorganism for enhanced shelf life. Development of flavor and texture, so it tastes better, you formed a certain texture that is desirable. Destruction of toxins, toxic compounds, or inhibition of inactivation of inhibitors, enzyme inhibitors, for example. Improvement of nutritional quality, because when we process, the protein becomes potentially more digestible if it is open structure, partially denatured. The starch can be digested without processing, without heating, without cooking. The starch is not digestible. So basically, processing is not a surprise, is required. However, it results in desired and undesired changes in protein functionality, such as, can anybody think of desired changes?
Like water holding capacity or gelation.
Right. So partial denaturation enhances gelation, enhances emulsification. So that is a good thing. Okay. What else?
Solubility?
What's that?
Solubility?
Excess processing can result in polymerization and undesired loss of solubility. Excessive denaturation and polymerization can result in reduced digestibility. So here's this slide. I don't know if you have that in your notes, but desired getting rid of enzyme inhibitors such as trypsin inhibitor, improve digestibility if the protein is partially denatured, not polymerized, and not large polymer, because that actually causes loss in digestibility. We talked about better functionality, emulsification, foaming, heat-induced gelation. And the undesired, we talked about solubility. If the protein is completely denatured and polymerized, it's going to be insoluble, fall out of solution. It won't be functional. So that is undesirable. Inhibition of desirable enzymes. let's talk about cheese, for example. When you're producing cheese, they heat the milk to lower temperature than usual so that they don't inhibit endogenous enzymes, because endogenous enzymes are needed for cheese ripening later on. So sometimes you do not want to inhibit these enzymes. So that's why it could be undesirable. Maillard reaction is in the middle. Why do I have it here? Because it involves proteins, reducing sugar and proteins. Sometimes it's desirable for color development and flavor development in baked products, and sometimes it's not desirable in forming browning and off flavor during processing. Okay.
I'm sorry for the previous slide. It's not clear for me the loss in digestibility.
Yeah. So when the protein is denatured excessively, and we'll talk about different types of denaturations and denaturing agents, then the protein might be, I lost the word. The protein can form polymers. And if the protein formed a polymer, that means protein and another protein and multiple proteins are interacting with each other, you end up with a big kind of aggregate. So that aggregate is very hard to digest, because then the protein, the digestive enzymes in our body won't have access to the different peptides to break all the peptides because of this large kind of aggregate. So then we lose on digestibility. So that's very important when we look at PDCAS, for example, and they say, oh, the PDCAS of this protein is 0.9. Yeah, but under what conditions? Processing impact PDCAS, protein digestibility corrected amino acid score. So that's why we need to take into account when we are determining PDCAS, we are determining that based on whether the protein is processed, non-processed, polymerized, not polymerized. It will impact digestibility.
Can we have example for that?
What do you mean?
Like certain food that could lead to this.
Okay, I can tell you, for example, the protein bars. So protein bars over time, they harden. So that's a very common phenomena with protein bars. Over time and over storage, proteins form covalent linkages, either via disulfide linkages or by Maillard reaction polymerization. And with that, you get this hardening of the texture. And not only that, then this protein that is now causing the hardness of this bar is also very poorly digested.
Thank you.
Mhm.
I have a question about cold gels. So when we
About
cold
what
gel-
gels? Sorry?
Yeah, the reversible gelling for
Yeah.
micellar casein. How does that work? So we do the cold gel, and then when we put it back to like 95 degrees in a water bath, you can get back it in solution, and then you can again gel it, like cold gel it again. How does that work?
Yeah, I'll be talking about that when we cover gelation. So what happens is when you, we'll talk about it in a few slides, but since you asked now, so when you heat, hydrogen bonding and electrostatic and van der Waals, they are exothermic. So they are, when you heat, then they are weakened. So then the protein is solubilized. And then when you cool, then the bonding starts forming again, and then they form a gel. So if you think about gelatin, for example, it comes from collagen. It's mostly stabilized by hydrogen bonding. So when you take collagen and you heat it, hydrogen bonding will be broken, and then you have the protein in solution. When you cool it, hydrogen bonding will start to occur, but they occur in a randomized way, not the original way, and then you form a three-dimensional gel. But then if you heat it again, it will be soluble again.
Okay.
All right, so this is reversible, but if you get covalent interaction happening, that is irreversible. Once a gel is stabilized by covalent interactions, that is irreversible.
Okay. All right. Thank you.
Pam, just a quick question.
Yes,
You were talking about how PDCAS, like you need to consider processing conditions.
yes.
Do you know, like I can't imagine that smaller companies that make a protein bar or protein shake retest PDCAS of like a novel protein. So, I mean, we have to assume that like nutrition labels aren't necessarily accurate.
They're not accurate.
right?
Yes.
Okay.
Yes, absolutely. This is an issue. This is a major issue. And the claim that they put on the label, this is very controversial because they really have to take into account once they process, they really have to take into account what happened to the protein digestibility. And if they don't, and they just assume that they're starting with soy protein, and soy protein has PDCAS of 1, but they're processing it. If they don't take that into account, then it is not real. And there's a lot of controversy around that issue.
Do you know if like any regulation is being talked about around that?
Not that I know of. It's kind of like up in the air. So no, I don't think there's a lot of complaints, especially that I know that producers don't want to open this can of worms, basically, because then they have to report the true value. And it's like reporting the protein using the conversion factor of 6.25 all the time, where it is not really 6.25 for some proteins, but they do use 6.25 to get higher protein purity. But there's no regulation around that either. So yeah, life is not perfect. Okay, good. Any other questions? All right, so protein denaturation, basically we talked about the formation of the protein tertiary structure and quaternary structure and stabilizing it. But we're not, what we're going to talk about now is the reversal of that. When the protein contains the free energy, it forms a stable structure. However, if there's any denaturing agent that's going to cause any breakage in bonds, that's going to cause the conformational entropy to increase. And once the conformational entropy increases, this is a negative here, delta G of denaturation equals delta H minus temperature by entropy or change in entropy. So when you have few bonds broken, you created chaos, more free energy and movement, and then you get a negative delta G, and we call this spontaneous denaturation when delta G is negative. Okay, so what happens when the protein is in its stable form? So usually in aqueous, your hydrophobic residues tend to not like to interact with water. If they are on the surface, the water molecules form kind of ice-like structure around the protein, basically. And the protein assumes its most stable form. And water is excluded from the hydrophobic regions. Other bonding occurs. So you have hydrogen bonding, ionic, hydrophobic, and between the residues and the water is excluded. And then the protein assumes a configuration that has the minimal free energy, minimize the free energy. So the one thing here is that what we've just been talking about is that the stabilization of the tertiary structure of protein is entropy driven, meaning you want to get rid of this chaos, and you contain this free energy by bonding until you reach to the most stable form with the minimum free energy. At low temperature in general, delta G is positive. There's nothing there that low temperature, no denaturants, there's nothing that's going to break any bonds. Then you have a delta G that is positive. You have stable protein, stable structure. There's nothing that will favor denaturation, basically. However, when we have high temperature, when we induce heat, when we have heat, we are breaking some bonding naturally, breaking hydrogen bonding, breaking ionic interaction, van der Waals interactions, and then there is an exposure of hydrophobic groups to water by means of unfolding of the protein. And with that, like I said, delta G would be negative, and the denaturation is spontaneous. So few broken bonds, chaos, delta G negative. So with the heat, we have increase in thermal kinetics. So facilitating unfolding, basically. And like I said earlier, the hydrogen bonds, electrostatic, van der Waals, these interactions are exothermic. So in the presence of heat, they are destabilized, whereas hydrophobic interactions are endothermic. So they are stabilized by heat up until a certain temperature, above a certain temperature, which is specific for different proteins. We'll learn about that later. Every protein at different denaturation temperature. Above certain temperature, the structure of the water around the hydrophobic residues breaks. And therefore, it destabilizes hydrophobic interactions. So at above a specific denaturation temperature, above a specific temperature for every type of protein, we even destabilize hydrophobic interactions within the protein. So how does amino acid composition affect thermal stability? That's the last question, and then I'll give you 5 minutes break. How do you think amino acid composition affect thermal stability? Anybody?
Is it with different amino acid sequences, you would have different types of bonding, and with different types of bonding, that affects how your protein gets denatured?
Exactly. So, for example, I always like to give this whey protein compared to casein protein. Whey protein is a very structured protein. It has a very ordered secondary structure that is stabilized by hydrogen bonding. So hydrogen bonding is sensitive to heat, whereas caseins are stabilized in a quaternary structure with a lot of hydrophobic interaction that stabilizes this structure. And we'll learn about it in more detail when we cover dairy protein. But the amino acid composition of the casein is different than the whey protein, giving it different secondary and tertiary structure. So that impacts how the protein is impacted by temperature. So for example, whey protein denatures around 70 to 75 degrees C. Casein protein denatures up to 140 degrees C.
I have a question. Sorry.
Yes.
Let's say, for example, that proteins that have more, like a higher quantity or more availability of non-polar amino acids will be more resistant to temperature for some. That's the idea then.
Yes. So when you have more hydrophobicity, and also there are other things that impact denaturation temperature other than the hydrogen bond stabilization versus hydrophobic interaction stabilization. You also have disulfide bond stabilization. So the more you have disulfide bonds as well, the more heat is needed to denature that protein as well. There's some the other factors that impact denaturation temperature. Yeah, but that all brings you to the amino acid composition. If you have high cysteine, you have high potential for disulfide linkages, for example. If you have high percentage of hydrophobic amino acid, you have high degree of hydrophobicity that stabilizes that structure, whether it is a tertiary structure or quaternary structure, such as the casein micelle is a quaternary structure. All right, I'm gonna give you 5 minutes break. So we'll be back at 3:55. Okay, let's come back.
Before we move on, Pam, can I
Yes.
ask a question about protein denaturation?
Yes.
So does the delta G equation explain for denaturation of proteins in freezing temperatures? Because I know ice can sometimes denature proteins.
So no, I won't say the freezing temperature itself is not the cause of denaturation. In that case, is the change from water liquid to ice structure that may impact the protein itself. So it's not necessarily the temperature of at which that protein is stored, is what happens when we, let's say, we have the protein in frozen conditions and then we thaw it and then we freeze it again. That freeze thaw cycle causes the formation of the every time ice crystals to form, and they expand. So the ice crystals are bigger in volume than liquid water. So they expand and impact the protein and gradually causes protein to kind of break some bonds there. So it's not the temperature, the freezing temperature itself that causes the protein to denature. Is what happening to the water and the amount of water too. Okay. So let's go back to where we were. First, I need to share my screen. Okay, so all right, so here's where we talk about water and impact of water on denaturation. So water impacts denaturation, thermal denaturation. So dry protein powders are relatively very stable. However, temperature of denaturation decreases rapidly as the water content increases. So if we look at this diagram down here, so we have water content on the x-axis, and we have temperature of denaturation on the y-axis. And here we have the enthalpy, change in enthalpy. So with a very low water concentration, you get very high denaturation temperature. As you increase the water, you can see that there is a dramatic decrease in denaturation temperature and accompanied by an increase, obviously, in enthalpy. A change in enthalpy is high. So what happens in the presence of water and heat? So you have thermodynamics going on. So the water is moving, right? And then the water hydrates the protein and partially penetrates into the cavities of the protein, causing swelling. So basically, when the water is in, it kind of breaks the internal bonding, and then the protein internal bonds are broken because then the water is there and the water forms hydrogen bonding instead of having hydrogen bonding formed, for example, between the residues within the molecule. So then it becomes a gradual or spontaneous, again, denaturation. So the more water you have, the more of the thermodynamics of the water that would facilitate breakage of bonds that are internal to the protein itself, and that will facilitate unfolding. Okay, so this is kind of a diagram that shows the denaturation of the protein measured in comparison to a denaturant. So denaturant can be denaturing agent, reducing agent, for example, or SDS, sodium diazosyl sulfite, or it could be temperature or pH. So, and then we're measuring here denaturation. We can measure it by increase in surface hydrophobicity, for example, or measure it in terms of a reduced solubility. That's why you see an increase here. If we're looking at surface hydrophobicity, for example, we can see an increase in that. Or if we're looking at solubility, we can see a decrease in solubility over time. So Y can be anything we are measuring to determine or assess denaturation. And it says here it is a two-state transition model. So because once few bonds are broken, then you reach to a point and then it kind of spontaneously, everything kind of breaks loose. So once few bonds are broken, the whole protein unfolds because there's enough energy to kind of cause chaos and break more bonds are broken, basically. If we want to think about denaturation as in simple terms, we can say it's simply unfolding of the protein. For example, if you have a globular protein, when it's fully denatured, it resembles a random coil. So random structure. Denaturation can be loss of quaternary structure. We'll learn later on a lot of proteins associate via quaternary structures. So you can have dissociation and association by changes in pH, salt, or temperature. So you can lose the quaternary structure. So that is one form of denaturation. You can lose the tertiary structure. That's another form of denaturation. Or you can also have denaturation at the secondary level. So oftentimes when we talked about FTIR last time. When we measure FTIR, when we measure secondary structures by FTIR, we look at alpha helix and beta sheet and random coil. And oftentimes we take the ratio of alpha helix to beta sheet as an indication of denaturation. Of course, if the protein is hydrolyzed, that's primary structure, denaturation of the primary structure. So if we have hydrolysis, also this goes under denaturation. Then there is the reversible and irreversible denaturation. So short-term action. So usually I bring to the class with me like a little elastic band. So if you take an elastic band and you just stretch it once and release it, it just goes back to its form. But if you just keep playing with it and expanding it and applying pressure for a long period of time, then it will become a little bit looser. So it won't be reversible anymore. So basically short-term action. So if I put, let's say, take the casein, for example, and I reduce the pH to the isoelectric point, they will form a precipitate, but if I neutralize, they're going to be soluble again. So applying the denaturant for a short period of time basically result in partial unfolding. Some of the native structure of the protein is preserved. And once you remove that from the stress, it just could discontinue to denature further, basically. However, if we apply heat or pH or any denaturant for a long period of time, so the protein unfolds, but not only that, what happens with the protein unfolds, and if we still have the conditions that enhances hydrophobic interactions, for example, then the proteins are going to come together via hydrophobic interactions because now they're unfolded, the hydrophobic groups are exposed to the aqueous, they don't like that, then they want to come closer to each other. And if I have disulfide linkage potentially formation, then this becomes irreversible denaturation. Also, even hydrophobic interactions alone can become too strong to be reversible without any addition of any agent to break them apart. So excessive heat Excessive denaturant can result in irreversible denaturation, mostly caused by strong polymerization. And of course, if the polymerization is stabilized by disulfide linkages, then that is the strongest form of polymer that can be formed and hard to break apart. Protein concentration plays a very important role. So when we look at proteins in solution, if we have 1% protein versus 5% versus 10% protein, the higher the protein concentration under denaturation conditions, the more chances that you're going to form polymers, because you have more of the proteins in the solution and you're heating them all together. Then you have more chance to form polymers than if you have a dilute protein solution. So protein concentration takes part in the formation of irreversible denaturation.
Pam, I have a quick question.
Yes?
For what's considered a long time for like if you're going to partially denature a protein?
Yeah.
How long is a long time before it doesn't renature at all?
Yes, see, protein is not very simple molecule, and every protein is different. So 1 minute for one protein might be just enough versus another protein, maybe 10 minutes. It depends on the protein type and the bonding that stabilizes that protein. So it really varies. It's not one shoe that fit all, basically. It really depends. And you discover that by experimentation more than anything. Okay, so denaturation and renaturation. So you can also denature the protein by breaking disulfide linkages. So you can add a reducing agent and you unfold the protein by breaking the disulfide linkages. But if you remove the denaturant, in this case here, beta-mercaptoethanol reducing agent and a urea, a detergent. So if you remove them, then the protein may go back to its original structure. However, if you induce if you add different concentrations, what this is showing you is at different concentration, you can get different results, especially with reducing agents. Sometimes reducing agents reduce the bonds, and sometimes a certain concentration reduce the bond and induce different types of bondage. So for example, here it kind of reduced it, and then with a little bit more of reducing agent, it formed different type of bonding. So it didn't necessarily go back to where it was. And if you have other proteins, high protein concentration in solution, you might form disulfide linkages with the other proteins. So reducing agents does not necessarily always reduces the protein. It might induce what we call disulfide interchange, where we get new disulfide linkages occurring that were not originally there. You can make the denaturation completely irreversible and avoid reformation of disulfide linkages by adding performic acid. So that will react with the sulfidyl groups, and then they're no longer going to form disulfide linkages, for example. So this is irreversible. You cannot go back anymore. So other denaturing agents, pH. So when you have like charges, so alkaline pH, acidic pH, very high pH, very low pH, will basically result in like charges. And when you have like charges, then you will have repulsion within the protein itself, within the actual molecule. Then you would result in swelling and unfolding. On the contrary, if you have the pH at the isoelectric point, then you form aggregates, such as the formation of acid curd that can be mostly reversible unless it sat there and all of the bonds settled and it formed a strong gel, then it won't be reversible anymore. Partial hydrolysis, so at alkaline or acidic pH, we might get deamidation. So asparagine and glutamine becomes aspartic acid and glutamic acid. And we can get sulfidyl groups, especially at alkaline pH. If we have three sulfidyl groups, they will form disulfide linkages at alkaline pH, which promotes oxidation. And then aggregation occurs, and this is irreversible denaturation. Salt. So we talked briefly last time, maybe, or I don't know if last time or the time before that, we talked about salting in versus salting out. So salting in really helps increase the charge load on the protein. So the salt reacts loosely with the protein on the surface and enhance water interactions with the protein. So this is, for example, we said in acidic beverages, they add phosphate, it helps stabilizes it helps stabilize the protein in these beverages. However, high salt concentration will basically shield the charges on the protein and prevent the protein from interacting with water, and then break internal ionic interactions. And instead of forming internal ionic interaction, they will form strong interaction with the salt, leading to denaturation, basically. So there are different types of salts. There are salts that stabilizes the protein, and then there are salts that destabilize the proteins. So if we look at this figure here, you have temperature of denaturation, and then you have different types of salts. So, and the anion salts impact denaturation more than cation salts. So for example, here, NaCl, sodium bromide. So if you see here, with higher salt concentration, you actually stabilize the protein, but a different type of salt, here's like sodium thiocyanate, for example, higher salt concentration destabilize the protein, causing the temperature of denaturation to be lower. So these two types of salts, we call them Cosmetrope and Ketrop. So Cosmetrope are like sulfate, phosphate, and fluoride of sodium. So what they are, they bind loosely to the protein and they enhance the solvent protein interaction. So this carrot shape is the solvent. So when you have Cosmetrope in solution, you enhance water protein interaction. And when you enhance water protein interaction, denaturation temperature will be higher. The other form is the Ketrop, which are different salts that bind very strongly to the protein, prevent protein-water interaction, causing the denaturation to occur at lower temperatures. So there are the two different types of salts, not necessarily just the concentration, but also the type of salt. So other K-tropes can be the sugar. So sucrose can be a small molecular weight compound that also stabilizes proteins, whereas guanidine HCl is destabilizing, is a small molecule that will interact closely, strongly with the protein and destabilize the protein.
Where does sodium chloride fall?
Sorry?
Where does NaCl fall? Is it a cosmotrope or a...
So NaCl, let's see here. So NaCl is this triangle. So it is a cosmotrope in this particular example.
Pam, do you mind just repeating again the is it a K? I forgot exactly how you just K-trope. Can you just explain that again? What it does with the protein?
Yeah. So the salt. So the additive. This is the salt, which is the black circle, will interact very strongly with the protein and prevent it from interacting with water. So when water is out of the picture, then the hydrophobic components can be exposed now, and internal ionic interactions will be broken because now you're having more ionic interaction with the salt. So therefore, your protein is less stable in this situation where you don't have net charge on the surface, basically, and you don't have water interaction. So that's where you have the least stable state. So that's at this point, it denatures at lower temperatures. The more you have interactions with water on the surface, the more you have charge on the surface and interaction with water, the higher is the denaturation temperature. Okay. This is an experiment we did back at Purdue in lab with students, and we looked at denaturation of albumin, egg protein. And we looked at temperature of opalescence, which is an indication of denaturation. So that at the temperature at which we see the cloudiness, okay, the white color, that's an indication of that the protein denatures. So if we just put the albumin in water, it will reach opalescence at 85 degrees C. But when we put sodium chloride at a particular concentration for that particular protein, again, every protein behaves differently with different salts. We did show on the other image that with sodium chloride, you get a higher denaturation temperature. But in this case, at that particular concentration, we actually reduced the temperature of denaturation. We reach opalescence sooner. But with ferric chloride, no matter how much we heat it, it remained clear. Why? Because ferric chloride forms linkages with sulfhydryl groups and keeps the protein from unfolding. So it won't unfold, it won't form hydrophobic interactions with another protein solution, so it doesn't give you the opalescence. When we add sucrose, it enhanced, but 0.1 molar versus 0.01 molar, it enhanced opalescence. So sucrose acted as, not enhanced, sorry, it increased the temperature. So the denaturation occurred at higher temperature with the sucrose at this particular concentration. With acid, at high molarity, we measured the pH, it was about 1.75. Very, very acidic. So you have a lot of net charge, then you don't reach opalescence. So it doesn't form protein-protein interactions because of the repulsive forces due to very high net positive charge. But when the acid was low, you have a pH that was closer to the isoelectric point. So you have less charge now. Then the denaturation occurs at lower temperature. So one thing to remember is with more charges on the protein, with more interactions with water and more charges, the higher the denaturation temperature. With less charge on the protein, less water interaction, the lower is the denaturation temperature. So that's what you want to remember. Okay, other denaturing agents. So organic solvents. So basically organic solvents are going to disrupt non-covalent interactions. So hydrophobic interactions are weakened. So basically, instead of the amino acids with the hydrophobic residues interacting with each other, in the presence of organic solvent, they want to dissolve in that solvent. So the protein opens up and allows that nonpolar residue to dissolve in the organic solvent. Hydrogen bonds are strengthened. And then electrostatic interactions are increased as well as electrostatic repulsions. So when you have low permittivity or, the current, when you have less water, So the current is less in organic solvent than in water. So you have more electrostatic interactions or repulsions. At high concentrations, all solvent cause denaturation, basically, due to solubilizing of the side of the nonpolar residues, as I just mentioned. Other denaturing agents, reducing agents, we talked about mercaptoethanol is a reducing agent, glutathione is a reducing agent, cysteine is a reducing agent. So it basically breaks down the disulfide linkages. We have urea, for example, guanidine hydrochloride. They bind strongly to the protein, and they form hydrogen bonding, and they kind of destructure the water around the protein itself. And sodium disulfate, they break down non-covalent interactions, they bind strongly, and they impart, it's kind of like a very powerful denaturant that would be irreversible denaturation in the presence of SDS. Yeah. Okay, enzymes. So, like I said, breaking the primary structure is also the denaturation of the protein. So the example here is the formation of the curd. So when you have add your chymosin, it's going to cleave off the glycomacropeptide that stabilizes the casein micelle. Then the caseins will fuse together by hydrophobic interactions, as well as calcium phosphate bridges. Physical denaturation also by beating the egg, for example, we're denaturing the egg proteins, and then they form a film around the air bubbles to form the foam and stabilize the foam. Kneading the dough. So when we are kneading the dough, what happens is that the gluten forming proteins denature, and they come together and they form strands that are linked by hydrogen bonding and disulfide linkages. Irreversible. Once gluten is formed, it's irreversible denaturation. And the beating of the protein, incorporation of air, we are restructuring the protein where its aqueous is towards the water phase, and the nonpolar is towards the air bubble. So basically the protein changes structure, unfolds, forms a film around the air bubble, and stabilizes the foam. When we talk about foaming, we will talk more about that, the structural changes of the protein as it emulsifies, as it gels, as it foams, etc. Okay, can we list few methods to assess degree of denaturation?
DSC.
DSC, yes. So other than DSC. Let me see, do I have, yeah, I have examples. Okay.
Whey protein nitrogen index for like the whey protein denaturation?
Nitrogen index. So do you mean solubility?
No, they actually determine the denaturation, like what is the difference caused in like processing whey protein. So they analyze the denaturation of whey with respect to that with the whey protein nitrogen index procedure.
So what do you mean with nitrogen index? So there's so many different meanings, and so they would look at the protein, they solubilize it, and potentially measure nitrogen in the supernatant and get you nitrogen index. Is that what you mean?
I think so. I haven't actually done the method, but I remember studying about it in dairy chem class, that they would do it for whey protein denaturation. So
yeah, so it's basically looking at solubility, changes in solubility. This is indication, and because we're going to talk about later how we measure solubility, but basically when a protein denatures, it will precipitate out of solution with minimal force, and then you can determine how much protein remains soluble, and then that usually you determine that by looking at nitrogen content. And then you can convert nitrogen to protein. So measuring solubility is one way of determining denaturation. So DSC measures the enthalpy and the temperature of denaturation. What else can you think of?
I think earlier you mentioned opalescence.
Yes, turbidity.
The beta sheet ratio.
Yes, yes. What else? We talked about a lot of different things. Yes. All right. Here they are. So basically, we said DSC, yes. FTIR, looking at the distribution of the secondary structure. Raman spectroscopy, we talked about it also last time. In Raman spectroscopy, it shows you shifts in banding upon denaturation. So you can see that you can evaluate the protein denaturation based on the shifts in the bonds and new bonds formation and removal of some ionization as well. So you can look at ionization state, you can look at new bonds formation, and you can look at shifts in different bonds as well. So here in this example, we looked at whey protein isolate without heat and with heat. We do see differences in the spectra. We do analyze the spectra and determine exact differences. And we look at, for example, partially glycated whey protein, non-heated and heated. If you remember, I told you that when we glycated the protein, it had structural rigidity that resisted denaturation. So if you look at the spectra, the Raman spectra here versus no heat and heat, you barely see any changes. So this indicates that this protein is much stable than it's not glycated form. And we look at impact of pH as well. When we heat at different pHs, we can look to see what happens to the structure. We see lots of changes in whey protein isolate, whereas the glycated protein, regardless of pH and heat or no heat, you maintain the same structure. So it's fascinating what we can do. Definitely we dive into what exactly changes versus not change, but that is definitely tells us about the tertiary structure of the protein and how it could change upon denaturation. DSC, we talked about measuring, it tells us degree, onset of denaturation and temperature at which the protein completely denatures. And it tells us the enthalpy, how much energy it took for that protein to open up and denature. If we don't see any endothermic peak, that means the protein has already been denatured. Measuring absorption, turbidity, looking at basically not turbidity per se here, but when a protein unfolds, it fluoresces more because now the hydrophobic groups like tryptophan, tyrosine, and phenylalanine, otherwise that would be inside of the protein because they're hydrophobic, would be more exposed. So potentially you have higher absorbance and fluorescence. Solubility test. So the more the protein is denatured, the less it's going to be soluble. And we'll talk about solubility tests later on. Digestibility, we can measure digestibility, and if the protein, depending, the digestibility may increase. That's partial denaturation. Decrease, that is complete denaturation with polymerization. If we have enzyme, we look at enzyme activity. If we want to look at polymerization, we can look at electrophoresis, gel electrophoresis. In this example is whey protein, beta-lactoglobulin, and alpha-lactalbumin. We heated it at 75 degrees for intervals of 5 minutes, 10 minutes, 15, etc. So you can see with every 5 minutes of treatment at 75 degrees, we lose the monomers and we start seeing polymers on darker polymers on top of the gel. That's with increased heating. So with increased heating, the protein is denatured, but we're also enhancing formation of polymers, especially through disulfide linkages. Okay, so with that, we're going to move next to protein functionality. So we have about 10 minutes maybe. I'll start the protein functionality. Stop sharing this one. Any questions about denaturation before moving on?
I just have a question. It's not about denaturation, but about unfolding. So
Yeah.
if a processing method is going to partially unfold the protein, so is it possible that both hydrophilic and hydrophobic groups are getting exposed? Or is it always only hydrophobic?
No, when the protein unfolds, I mean, when you have your tertiary structure, it doesn't mean it's not in perfect situation that all of your polar proteins are on the surface and all of your hydrophobic proteins are inside. You do have a balance in distribution. So you do have some polar protein amino acids that wind up in the interior moiety of the protein interacting via different types of interactions. So it's not, unfolding is going to expose new groups, but mostly your hydrophobic groups are mostly in the inside, right? So these will be mostly more exposed than usual. But sometimes the protein in its native form has hydrophobic surfaces, such as, plant proteins. Soy protein has very high surface hydrophobicity. Even in the native form, you have you have high percentage of hydrophobic amino acid. They would tend to be on the surface. Some of them would tend to be on the surface, and then the water forms that crystalline shape around it to keep it stable. Okay. All right, so let's share my screen.
Your voice is a robot.
Is it?
Yes.
That means I have to stop recording, leave, and come back.